All right, ladies and gentlemen, good afternoon. My name is Robert Luna, and so today we're going to be talking about Chapter 18, which is the molecular techniques for epidemiology and the evolution of arthropod-borne pathogens. So in this chapter specifically, we're going to be talking about a lot of how different methods when it comes to epidemiology and tools on how, in various situations, how pathogens are detected in vectors and so forth. These tools and principles are critical to how we can see the development of these, arthropod-borne pathogens or diseases, and how pathogens can possibly adapt to their hosts as well towards the end. Okay, and so let's go ahead. So when it comes to the definition of epidemiology, the actual meaning of it behind it is, like it says there, the study of patterns and the determinants of the spatial and temporal distributions of a disease. And that's the key right there. The study of the patterns and the distributions of a certain disease from a specific pathogen on a vector. Okay. And so when it comes to, again, this chapter, right? We're going to be talking also about genetics, and we're going to be talking about biochemical markers specifically to identify pathogen species and vectors. Specifically, when we talk about different methods like PCR or electrophoresis, what they use specifically to identify pathogens in vectors. Okay. Now, in the very beginning of this chapter, um, we talk about how the basic reproductive ratio, like it says here, the equation R sub 0 equals B times K times D. Now, each one has its own meaning. Real quick, I'll mention it. B is the infectivity rate. K is the number of potentially infectious contacts the average infected insect has per time, and then you have D, which is the multiplied by the duration of infectivity of a particular insect. So basically, you have this is one way of calculating this ratio, but there's another way. And another way is using the average lifespan of an arthropod vector in the population over the average age at infection. So throughout these next few slides, we're going to be talking about specifically a few equations as far as how we can calculate specificity or sensitivity. There we go. I was trying to remember the other one. So, but interesting to note that again, there's these two equations, and that we'll talk about kind of later. But there's also different versions of other equations that we'll talk about later in this chapter as well. But again, this is just for, and I did forget to mention, this is the closest approximation to the answer as far as R sub 0. Now here you can see 5 types of taxonomic groups shown here. You have arboviruses, you have bacteria, you have rickettsia, protozoa, and nematodes. For the most part in this chapter, we talk about mostly the first 3. Protozoa, very little, and nematodes, practically none. But for sure, the first 3 are mentioned quite a bit. Now, real quick, I did forget to mention, going back real quick, if it is less than 1 on one of those equations, specifically the second one, every generation of infection of an insect won't have the same effect as the previous generation did. So what that means is another way of saying is that if R sub 0 is less than 1, more than likely that population of pathogens will probably die out, more than likely for sure. If R sub 0 is going to be greater than 1, that means that population of pathogens is going to be surviving at a given point in time. Now, in molecular epidemiology, we tend to see different patterns or peaks of an emerging pathogen, and it helps us understand, the behavior and adaptation of these pathogens. So specifically, when it comes to the pathogens, how do they do that in vectors is what we'll also see when it comes to these taxonomic groups here as well. Now, in the right side, you have a table, and it's table 18.1. You have the current molecular techniques for detecting pathogens in vectors, all which you see in each taxonomic group. Mostly you'll see reverse transcriptase PCR and so forth. But for the most part, it's PCR. And we'll talk about briefly as we go through the chapter here. Okay. Now, in order to get the best results, you have to make sure that whatever technique you use, either reverse transcriptase or PCR, you have to make sure that the technique is going to be valid and available. Otherwise, you might end up with false positives or false negatives in some cases. Now, when it comes to testing, you have two types. You have screening tests and diagnostic tests, and that's what we have here. Screening tests, can help with early detection of a disease from a vector. Diagnostic tests can help confirm what the screening test revealed. The good thing is that both of these tests, however, have the same goal, which can try to get the best accurate result as possible. Now, whenever you have organisms, though, this is actually an interesting thing here. Whenever you have organisms that are going to be known or maybe suspected that they have a certain disease that is going to be like at a higher prevalence, this is going to be termed a high-risk organism, which basically has a disease that can rapidly spread through different vectors or organisms in general. Now, when it comes to, let's say you're testing one of these, you're using one of these tests, and you're trying to determine the accuracy, there's two types of accuracy. You have sensitivity, and then you have specificity. Sensitivity is the probability that a test will correctly identify infected organisms, whereas specificity is the probability that a test will identify uninfected organisms. And those two things are very, very important when it comes to false positives and false negatives, because again, you can get those things. And so you got to make sure that when it comes to specificity, you can determine which organisms are going to be uninfected and which ones are infected, especially in sensitivity, like it says there, right? Now here in figure 18.1, we have a few variables to mention. Real quickly, D plus means the disease is present, D minus, no disease is present. On the right side, you have the total for the T plus and T minus, which basically means that the T plus and T minus, they're test results that are determined by screening or diagnostic tests. And as you can see in the figure, you have the numbers for disease and non-disease vectors, along with the total numbers being experimented. And you also have the T plus and T minus, as you guys can already see. And at the very bottom, you have the total disease and non-disease. Okay, so for the next few slides, you're going to be seeing the equations that you can use to determine, let's say, for example, true prevalence. We have apparent prevalence, accuracy, misclassification. And all these equations, you're basically determining the proportion of how many in general, and broad general answer, how much is a disease prevalent, or how much is there the disease present, the sensitivity of the disease, the specificity of the disease, and so forth. So true prevalence is the proportion of organisms tested that were diseased as assessed by the gold standard. Apparent prevalence is the proportion of organisms that are positive in the screening test. So using the numbers that we saw here, 18.1, you can use the D plus, which I have here, over the N. So the N is the total amount as a whole. So it was 149 as the total amount, and 33 was D plus. The disease was present in the organism. So 33 divided by 149 is going to be 22%. And you can do the same thing with apparent prevalence, T plus divided by the total, which in this case, the T plus was 96, divided by 149, you would get 64.4%. And so throughout the slides, you're going to see that, okay, there's different equations for different types of scenarios or proportions that you want to measure, and you get a certain percentage. And so misclassification, again, you have your equation here. Misclassification is a proportion of those organisms incorrectly identified as diseased. And this time you have false positives and false negatives as two variables divided by the total. And so here, you basically get the total. You will get the total of the positives and the negatives, which was 53 divided by 149, and you get 35.6%. And so you continue there. I did briefly mention I did include the numbers in there along with the picture there, so you guys can see what I'm talking about as far as the numbers. Sensitivity is a proportion of truly diseased organisms that test correctly, whereas specificity is a proportion of the non-diseased organisms that the test identifies. Okay, and so again, you have the true positives and the false negatives, and the true negatives and the false positives involved in both sensitivity and specificity. Okay, Now, going back to true prevalence, another way to calculate it is by involving the variables specificity and sensitivity, along with the apparent prevalence. So earlier, we calculated the apparent prevalence and the specificity and the sensitivity in the previous slide. So all you have to do is convert those percentages to decimals, and you would go ahead and plug it in, and you would get 37.5%. Now, proportion of false positives and negatives, again, they have their own equation as well. False positives are the proportion of truly non-disease vectors that the test misidentifies as positive. False negatives are the proportion of disease organisms that the test misidentifies as negative. And so they have their own variables there, false positive and true negative. And you have the percentages as well. 60% for false positive, and for false negative, 68%. Predictive values, positive and negative, they also have their variables there. There are multiple ways to calculate predictive values. You have the positive and the negative predictive value. Below you have both equations. Again, the variables are there, the numbers are there as well. 18 divided by 33, and then you have the 67% for the negative predictive value. Okay, so go ahead and continue. And of course, you have the equation there, predictive value with sensitivity and specificity as variables. And so you go ahead and plug in those numbers, and the next slide you have the actual answer with the numbers plugged in, which is 99.9%. And so here in this figure right here, we have 18.3a and 18.3b. And I'll just mention real quick, 18.3a is going to be considered a poor test compared to figure B being a good test. And the reason why is because your sensitivity value. The sensitivity value is going to be 51% in sensitivity when it comes to figure A, and figure B, your sensitivity is going to be 98%. So as you can see, the closer you are to 100, the more better or accurate test results you're going to have. And you can also see through the graph that there's a big overlap in figure A compared to figure B. And so that's another reason why, or that's another way how you can see that figure A is going to be poorly tested compared to figure B. Excuse me. When it comes to sensitivity and specificity, if specificity is increasing, positive predictive value is going to increase. Therefore, getting the chances of a positive result increases as well. However, if sensitivity increases, negative predictive value is going to increase. Therefore, the chances of getting a negative result is going to increase, which is why you see both graphs slowly, progressively going up. And depending on the overlap or the sensitivity values or the specificity values, they could be considered a good test or a poor test. There are a few scenarios or situations where you would want to see or use sensitivity or specificity along with your predictive values. Now, real quickly, mention this, series and parallel. Series happens when the result of every test run must be positive or the vector is negative. The parallel testing happens when the result of every test run is negative or the vector is considered positive for the disease. So it's kind of basically the other way around, the reverse, right? And so now that we're talking about culturing, when culturing or dissecting a vector, it could be possible to identify the pathogen. The only problem is it's going to be very, very difficult. The pathogen could die whenever you're trying to capture it, the vector itself. There is no method of culturing a new pathogen. An example of that is the sine nombre virus. I'm not sure if I pronounced that right, but it was mentioned in the book that it was detected by using a reverse transcriptase in PCR, and determining the sequence analysis was done before techniques were even established. So that was another reason why it was kind of difficult to get the pathogen. Another way is that if there is a new pathogen, it means that the culturing process is going to be very slow, and adaptation is also a big factor when it comes to certain pathogens. Especially when it comes to changing the genetic variation or the genome of the pathogen. Okay, so there are multiple ways of detecting arthropod-borne pathogens. One of them is going to be using multiple lens electrophoresis and PCR. So both of these have their own processes. It involves growing the isolate to higher so basically when it comes to MLE, multiple locus electrophoresis, it uses the genome, the nucleic acids, to kind of identify what organism it's going to detect. And PCR is kind of the same thing, but it uses specific primers called oligonucleotide primers. Okay. Now, the reverse transcriptase PCR and multiplex PCR. In reverse transcriptase PCR, they have arboviral RNA transcripts present, whereas multiplex PCR is a process that helps detect multiple pathogens by using multiple primer pairs to detect a different pathogen. Okay. Um, okay. Let's go and continue. Pathogen adaptation. So again, like I mentioned earlier, pathogens can adapt to whatever vectors that they try to bind to or bond to. So it depends on really their immune system. Pathogens need a weak immune defense from the host. So if the pathogen somehow does bypass through the immune system of the host, more than likely it's obviously going to be bonding to the host and it's going to be infecting the host. And when it does that, it's able to adapt and go through what's called co-speciation. And co-speciation means that the pathogen's phylogenies, in general, when it comes to the general definition of it, are going to align or parallel with the host phylogenies because the pathogens haven't infected the host yet. So once they do infect it, it's going to, the genes of the pathogen in a way are starting to align with the host. Vectors can reject or fight the pathogen through multiple ways. For example, through the feces or through the reproductive system of males and females. And for the most part, that's pretty much it. Those two ways that the host can reject the pathogen. And this is an example here mentioned in the book, like the symbiont relationship between microorganism symbiont species and the Cryptocercus roaches. Some of these vectors can reject the pathogen, like I said, through multiple means. And this is showing you again the phylogenies of both of these organisms, and slowly but surely they'll start to align later on. Now, when it comes to the current research, surprisingly enough, there is a paper out there that was published, and the research was done in Colombia. And so in La Mesa, specifically in Colombia, there was research involving multiple vector-borne diseases like dengue, the Chagas disease, and what they were, as far as the methods, what they were doing, they had multiple mammals being sampled, like rodents and bats, along with certain dogs and opossums. And when it comes to mosquitoes, DNA and RNA were extracted by using, like it says there, the ZR tissue and insect DNA mini prep kit or the quick RNA viral kit when it comes to the RNA. And for both, they used the quick DNA RNA viral kit to detect pathogens in general. PCR was another major method that they used to detect pathogens in these mammals. Okay, so now throughout the rest of the chapter, it talks about how different pathogens adapt and evolve in certain hosts. For examples of that are like the arboviruses, the plasmodium, filaria, the trypanosomes, and the leishmanias. They each have their own versions of adapting to their host, and depending on the host, depending on the immune system of the host, they could reject it. And so, and this is the reference.